Herkese merhabalar. Hello everyone. We are in Istanbul at Florence Center for Weight Management and Metabolic Health. Today we have a very distinguished and esteemed with us, Professor Mazhar Adli. Welcome, Dr. Adli.
Thank you. Thanks for your invitation.
So thanks for being with us today. So he's coming from Chicago. I would like to introduce him first. So Dr. Mazhar Adli is a full professor with a prestigious endowed chair position at Northwestern University, which, as we all know, it's ranked at the top 6 national universities in the United States. And Professor Adli is graduated from ODTÜ from Turkey and then started his academic career as a PhD student at University of North Carolina at Chapel Hill at UNC Medicine. And then he did his postdoctoral training and research at Harvard Medical School, at Broad Institute of Harvard and MIT, where he actually established a very specific epigenetic method, a nanochipstick method. And then he established his own lab at University of Virginia. And then with his huge success, then he got transferred to Northwestern University. So Professor Adli has been involved in many large-scale consortium-level projects, including the Human Epigenome Project. And indeed, he was the author of the first major human epigenome mapping consortium paper. So Dr. Adli has been awarded with numerous awards, just to name the few, like the V Scholar Award, Remont Scholar Award, Dean's Research Excellence Award, and last but not least, in addition to many NIH grants, that he got more than $25 million. He recently won the prestigious consortium award of more than $10 million, where he's developing technologies to reveal what all human genes do. So he's currently the chair and steering committee member of the NIH Morpho Consortium, and he's director of Genome Engineering Center at Northwestern University. So his laboratory is developing novel technologies to understand how the human genome and epigenome work together to function. So he's one of the world's leading expert in human genome and epigenome. And that was the reason that we invited him. And his laboratory was the first in the world to develop technologies to alter the human epigenetic information in a targeted fashion. And he published more than 70 research papers, which papers has been cited more than 25,000 times with this very impressive CV. So we are really honored to have you.
Thank you. Thanks for your invitation. And it's a pleasure to be here. Look forward to have this chat with you.
Thank you. And we always start with our question first. You know, we established this metabolic health center in the region in Istanbul, and so you saw the center. So what do you think about the Flowell?
Well, first of all, I think it's congratulations. I think it's a big success to bring all this team together and set up this comprehensive center. I think we know that now the living a healthy requires a comprehensive assessment, and it looks like you guys are doing exactly that. So congratulations, it looks impressive, and I want to congratulate the entire team as well.
Thank you so much, thank you so much. So we will start from the beginning and basics. So what is epigenetic or epigenomic?
Yeah.
So this is a fancy word that everybody wants to talk about or use it more than it should, but we really wonder what is epigenetics.
So as you said, I think a lot of people are using the word epigenetics and maybe few epigenomics. I think this is one of the words that are being used a lot, is misused a lot, and I think also abused a lot. It looks like anything when people want to cover something, they just throw the word epigenetics and throw a lot of things under that carpet. So epi in Greek means above, and genetics means the study of DNA. So anything above the genetic information, we say it's epigenetic. So because when we say anything above genetics, there are many things that go into that category, and we all call them epigenetics. But in essence, what it is that you can consider it's like a software of our genetic information. Every cell in our body has the same genetic information, but yet we have more than 200 different cell types in our body. So the obvious question, scientific question and developmental question that bothered many big minds was, how come the same genetic information can lead to all these different cell types? If they all have the same information, why do we have a skin cell so different than a brain cell, and that's so different than blood cell, for example? So that mechanism that dictates when the genetic information should be revealed is epigenetics. So And this was why I was interested in this topic. When I finished my PhD, I wanted to study this information, but at the genome level. So what does that mean? Our genetic information in every single our cells, if you unwind that DNA, it's literally 2-meter long DNA. And this 2-meter long genetic information is stored inside the nucleus of every cell. A brain cell uses different part of this information. A blood cell uses different parts of this information. And the mechanism that tells the brain cells that you should use only this part is epigenetic. So what is the source of this information then? The epigenetic information is stored in two main kind of compartments around the DNA. One is the DNA methylation. This is the most widely studied one, DNA methylation. It is a specific chemical mark on the DNA. And whenever this mark is, if it is in the certain region in the DNA, and we call promoter, the beginning of the genes, it usually tends to silence genes, means that that gene is not going to be open. That gene is stored for later. There's additional layers of information in the epigenetic information that is stored in the proteins that the DNA is wrapped around. We call histones. DNA is never naked inside the nucleus. It's always wrapped around this protein we call nucleosomes, histones. So the histone tails undergo hundreds of different modifications. So all in all, there's hundreds of different epigenetic information, epigenetic modifications around the DNA. The most widely used one and the easiest one, that's the reason why it's most widely used, is the DNA methylation. So whenever people say epigenetics, they only talk about the DNA methylation. And we should also highlight that even the methods that are used for this epigenetic, this single mark is not super cutting edge. So the bisulfite conversion even is not studying the entire DNA methylation because there's not a single modification on DNA in terms of just the DNA methylation. But we can go into detail later about that one. So that's the epigenetic. Any information above the genetic information we call epigenetic. There is also the word epigenomics. We tend to use this, this word to describe all entire epigenetic information in a given genome. The genome is the total genetic information in any given organism. For humans, we have 3 billion letters of genetic information. So epigenetic means that anything above this 3 billion letter of information is epigenomic information. So we started by saying that we have 200 different cell types, which means that we have 200 different epigenetic information in our body, basically. So when we talk about epigenetic, we should also highlight that it's a very cell type specific information. So the study when you study blood cell, it was different epigenetic information than when you study muscle cell or a brain cell, for example.
It's very interesting. So if I understand correctly, so when we say one word, actually we talk about there are 200 different cell types, has 200 different epigenetic information, and we are in the clinical world, we like to translate things fast enough so that we can use for humans or for diagnosing or doing risk assessment or doing interventions. But we are almost looking to the blood. So we are basically almost doing 5%, even it's not going to be fully just percent, but 1 out of 2 other cell types is just blood. So we are just looking a small part of the epigenetic information, and we're just generalizing that information to everything. And
exactly.
So okay, I'll get into the detail of that part. That's one of the
limitations of it, exactly. And even the blood, as you know, we have so many different blood cells. Even a T cell will have a different epigenetic than a B cell. But because it's easily accessible in the clinical world, the tissue, whenever they say human information, is typically a blood cell, because it's the easiest one that we can get.
Yeah. So I think I would like to get a little deeper in that, because the main trend now in the world is when people try to show something different, or even to say they're doing something different. They're saying, for example, for us, the nutrition really matters, and we know that so certain type of right health habits or the wrong ones, if I understand correctly, can change this information, which can reflect to certain good things or the bad things.
Yeah.
But how it's utilized, when somebody says epigenetic nutrition, or I'm gonna assess your epigenetic information, I'll tell you how to eat, or take this supplement, or take this drug. So it's a little over utilization, because they're simply just taking the blood, which is like it's a mixture of cells, it's a mixture of information. We don't know which one reflects or kind of which cell types or which is more dominant, but we are just making some report that we're thinking it's your body, but it's just certain cell types, and representation we don't even know. It's going to be different from person to person. And then what I see there, they're looking for certain mutations, and then they're saying if you have this mutation, you got to take this more. But if I understand correctly also, mutation is not about epigenetics either.
Yeah.
So it's a genetics, genetic information, and you can get it from a blood cell because that information is same for everybody else, but you basically wrongly label it as epigenetic, and you basically tell the patient that I found a different information and I overly generalize something.
Yeah, no, exactly. As you summarize, I think that is where the epigenetic word has been misused and also abused in sometimes, because it is a cutting-edge research, and as someone who devoted, big chunk of his career to this field, and I developed the methods for this one as well. It is good to see that the clinical side is catching up. It is trying to use this information, but it's also sad to see that it is being used as a marketing gimmick and marketing tool only. And when something becomes marketing tool, people make money out of this. And when they make money, there's a conflict of interest goes into it. They don't pay as much attention to the scientific basis of these things. And as you described, if you are making a lot of money out of this one, you don't even need to know what is really epigenetic. What are you actually measuring? And I know even people that I personally know who has never done an epigenetic research, and I know their background is not epigenetic. They're on TV and promoting these kits and these things for epigenetic coaching, epigenetic things, which is the sad part of the thing. What we need to know is to understand that epigenetic field is still evolving. We still need to map all this information. When I started my career, we were mapping this in human cell lines. Then we decided that we need to also map all these 200 different cell types. We actually need to create those maps. The epigenetic information, unlike genetic information, is malleable, which means that it can change over time. So there is a temporal aspect of it as well, which also comes back to the clinical use. For example, one is it's a cell type specific. Every cell has a different information. Second, even within the same cell, the epigenetic information that you measure now is going to be different 3 months down the road. And it is going to change with the environment. I think that part is why people are excited about it, because they market this one that, okay, if this is your epigenetic information, you should eat this kind of things, and you're good. There is no such thing that you can change the epigenetic information in our entire cells. You can change it, but we don't know, is it causal? Is it going to be better if you change entirely? Because this epigenetic information, there are some readers that read this information. There are some writers that write this information. Even if you feed someone with excess amount of certain methyl groups, these readers and writers control this information. And you need this control. Now, coming back to this, when they call mutations, I don't know if they are correctly using even the word mutation there. Because mutation, in order for something to be mutation, it needs to be very rare in the human population. But as far as I know, they are measuring some variance in the human population. And that part also is called genome-wide association studies. So they are associating certain traits, certain phenotypes with some genetic variations. These are very common variations in the human genome. For example, between me and you, there are at least 10,000 different genetic information differences. That's what makes me unique and what make you unique. And certain number of this variation can be associated. Again, the word is associated. It's not causal. Can be associated with certain traits. And these people are using and abusing this association as a marketing tool again. Because they say, okay, we will do this GWAS, and if you have these variants, here is your separate set of diet. And there is no such thing as proven way of building a diet based on just the genome-wide association studies.
Yeah, I think this is very important part. So, and I can tell this is the mostly abused part in clinical world or in the basically dietary tools or nutrition field, because every field wants to evolve, and we know how great job you guys are doing and all this epigenome research, epigenetic research. But what I understand now is simply when I look at this, like those reports, and that comes to us, like the patient comes to me with like 100 pages of a PDF, and give it to me like, okay, this is my epigenomic or epigenetic. I got tested, so tell me, what that means. And it's very discouraging for patient. At the same time, spending thousands of dollars, giving a blood, and comes a huge amount of information, which when I look, it's mostly genetic mutation analysis or GWAS, association studies, and it kind of say, oh, don't eat this and don't eat that. And then I had many people, I said like, I'm gonna pay it, so let's test it again. And the same person get tested with like 6 months later without doing anything. And then it just gives a different type of, and then say this time, like drink this. The previous one says don't drink that. So, and I see it's based on certain algorithm, which is not scientific. So there's no such a thing that, okay, eat this fruit this much per day, that will change your epigenetic that much, which is good for you. So as of, if I understand, it's not proven. So where we try to get is it's possible that your nutrition can change your epigenetic data. It can be good, or nutrition deficiencies or certain excessive nutrients or excessive vitamins or minerals can change this, but we don't know exactly in which cells and at what level, what dose, and what kind of changes happens. We just know that it can change, and we are just speculating based on a GWAS or variants or certain mutations that, and then the people just get overwhelmed with that report, or somebody's just using it and eliminating millions of things from people's life, and people just feel like they lost, and they're actually losing trust to new information, to modern science and medicine. And at the end, it's not actionable most of the time. Of course, we say, if you have these deficiencies, of course you need to improve. We don't like deficiencies. We try to normalize things. If somebody thinks if something is excessive, you need to bring it down. If you're deficient, you got to replete it. But it's not like just eat this and it will fix this, or it's the other way around.
There's not going to be a pill saying, okay, here is a pill and it will solve all your problems. Still diet, we know that is very critical, very important. Exercise is very important. I think none of these epigenetic molecules, things are going to beat exercise or your daily diet. You have to control those things. And I think if for our audiences, I want them to understand it like 3 words. One is, it's cell type specific. So not that you have one epigenetic information and that's it. All your body, in the cells in your body, have different types of epigenetic and epigenomic information. The second, I think, this epigenetic information is temporal. It changes over time, meaning that the thing that you measure today is not going to be the same thing as you measure 6 months down the road. So that information is changing. I think third is that the word epigenetic is misused a lot. So the field is still evolving, and one day we will have this thing, but the third critical part that I want them to understand is that we don't know whether it is causal or not, right? So there is an association. Association is different than causality. So we can measure things, it is there, but is it there because, like, this is exactly the question that we were asking when we were measuring this epigenomic information. This is exactly the reason why when I set up my lab, I wanted to understand this. We measure that, say, if a gene is silenced, certain epigenetic information is not there. And if it is active, certain information is there. Now, is gene is silenced because of the absence of certain things or presence of certain epigenetic marks, or is this epigenetic mark is absent because the gene is active?
So chicken and egg situation.
Chicken and egg situation. And so therefore, even if they suggest that what your epigenetic information is low in this sense and we should feed you with this, but other parts of the genome needs this information as well. So you cannot change it too much in a way. So half of your genome, you need to always silence certain genes and activate certain genes for any given cell type. If you try to activate everything, it's bad. If you try to silence everything, it's bad. So you cannot even play with this information too much. And that's also dangerous, to try to like massively change the epigenetic information.
And we know that certain things is even the same molecule is like palmitoleic acid that we discovered. We think that it's one of the rescue mechanisms of obesity. It comes out of fat tissue, but the same molecule activates one enzyme. The same enzyme activates in fat tissue, but it silences and inhibits the same enzyme in liver.
Yeah.
So the same enzyme is problematic in liver, is beneficial in fat tissue.
Exactly.
So what I understand is even from that, forget about the genetic, it's just biochemistry is cell specific, which means for our audience, it's very important. So the fat cells versus liver cells versus brain cells versus neurons, like they're all created differently. They have a different program to follow. They have certain enzymes or proteins or genes, like they're same information they all have, but they act differently based on those informations or which parts of the genome is active, that you cannot just alter with one molecule or just with one diet or one exercise or one pill or one medication. So
even like regarding to this one, I want to add this even more striking. The same chemical molecule, it's a methyl group. It can be added to lysine positions that are 4 amino acids apart from each other. It's exactly the same chemical molecule. If it is on lysine, histone lysine 4, for example, position, it has a completely different biological outcomes versus if it is on lysine 27 position. So one is completely activating gene, one is completely silencing gene. But it's the same chemical group.
So same chemical group, same amino acid. So lysine is amino acid.
Yeah, same on the same lysines, same histone tail. That physically they are very close to each other. It's the same chemical group. In one case, it is activating gene. In one case, it is silencing gene. And when you map this information, that's what we were mapping. In the entire genome, you see that all the active genes has this lysine 4 methylation, trimethylation, and all the silent genes have lysine 27 methylation. So you can give as much as methyl group as you want, yet it can silence certain genes and it can activate certain genes. So that's why it's very complicated field that it's still evolving, and we still need to understand, is it really causal or not?
Yeah. So it's very exciting. So I don't want to give the wrong message in a way that, so it seems like the science is really advancing, and where we try to come is to say, hey, if you have these mutations, so you're at risk for this. For example, like if you have this mutation or loss of function mutation, so you have tendency to have blood clotting. So you gotta be careful moving more, or you gotta use blood thinners. So maybe, or in cancer, it's totally different. There are targeted mutations that opens the door versus close the door. You can use certain targeted medications. It's different. So for precision medicine, we try to map all this information that clearly this is the target, But the biggest enemy of this big scientific target is prematurely selling this commercially to the audience, to patients, without reaching there and act like we already reached there, and then prematurely just get the blood, forget about all the cell types, and say, this is your recipe, and do this, do that, like all non-actionable information. I think we have to be careful about this and not to get discouraged and not to lose the trust to science, because we already advanced a lot. But
definitely, I want to also add exactly to this point that if a situation is a genetic mutation, like in cancer, there are a lot of these epigenetic modifiers that are being mutated in cancer as well. So in those cases, one of those readers or writers or removers is not there. So when it is not there, there is an excessive amount of accumulation of certain repressive marks or activating marks. Then you can use small molecules to inhibit those other enzymes to correct, to balance the situation. So in cancer, we already know that some of these epigenetic inhibitors have been approved by FDA and are currently being used to treat cancer. So in cancer situation is different, because there we have a mutation. In a way, the analogy that I would like to use maybe is like a traffic. There is a huge truck that rolled over, it's blocking the entire highway. And in that kind of situation, removing this truck, at least in partly, may correct this flow of this information. But in a normal traffic, it's very, very inefficient, and we still don't know how these molecules are going to affect.
So then, because the cancer research also cell-specific research, so you're working on pancreas cancer, you know the pancreas cell, or you're working on liver cancer, or like blood type or blood cell cancer, leukemia, that it's, what is changing, and you know what is causal, and you know how we can modify it. So that field is more advanced, versus more when things are normal, you have certain type of nutritional deficiencies, or you are on a Western diet, you're gaining weight, you're losing weight, you're not in a very ideal lifestyle. So how much of the epigenetic data reflects why it happened or how we can change it, it's not known yet. But that's being researched, and that's where we try to reach, but we're not there.
We're not, the field is not there yet, yes.
So one of the things that we are utilizing is, some of the big scale clinical trials also using the biological age. So now we know that the chronological age is not the biological age, because somebody's really like drinking alcohol, smoking, and really getting the processed food, and keep gaining weight and losing, all this weight cycling, like somebody has autoimmunity, there's already an inflammation in the body, is aging differently than somebody is really on non-processed diet, or have a little bit maybe better genetic advantage and better diet and better lifestyle, exercising daily, not smoking even at one time. So we know the aging happens in a different pace, and in clinic we like to measure things.
Yeah.
But what I understand from all this also, this is one of the at least best associated markers or regions that we know that associated with aging differently. So when we do biological age studies through the DNA methylation and adding some certain markers to increase the kind of accuracy of that measurements, so we are kind of still, we're not saying, we're saying a number, but we are simulating based on and more established associations.
Yeah.
Am I correct?
Yeah. I think DNA, this epigenetic clock, based on DNA methylation, probably has one of the strongest scientific bases, at least, because the correlation is very strong. The correlation, for those who are more statistically oriented, the R-square value is more than 0.9. Means that the correlation is very, very strong. Your age versus the epigenetic aging. We should also know that this information is being used. There are methods to measure those things, and you can measure in multiple different tissues. You can measure in blood cells. You can measure in other tissues, and each tissue has different sets of these regions of the genome that are associated, but the association is definitely very strong. We can use this one for certain things, but we should be careful that this is an association, but it's very strong correlation. I think the exciting thing is, since epigenetic information is changeable, now there has been a small-scale clinical trial with just nine people, that certain drug treatments and certain exercises may actually change this epigenetic clock and make look people younger with that clock. Now, we don't know the long-term consequence of this one, but that part of data, I think it's exciting. That seems like by interfering with the diet, with treating with certain chemicals, maybe you make the person look younger with the clock. And hopefully, in the long term, that will also translate into actually the overall wellness of that person.
Yeah, that's very exciting. And so far in metabolic health, so what is getting established is, so simple message, because everybody asks like, what supplement should I take? And these are all expensive supplements, and there are many of them, and people are going with an like question, and somebody wants to answer, because as a provider, you want to help. Normally our first principle is do not harm.
Yeah.
Primum non nocere from Hippocrates. Like it's, but sometimes people want to help more. That can cause like we are actually forget about the first principle, and then they're just, throwing 20 supplements there. And then, but what we are understanding is, if something is deficient, it's bad for you, simply. And then if you replete that to normal levels, it's good. But if something is normal, adding on top of that is not necessarily a good thing. So that's where we have to be very precise and very careful. And from that biological age, so in endocrinology field or nutrition field, through those measurements, what we found, when you supply multivitamin simply, or when you supply like iron to people who have iron deficient, or vitamin D to people who have vitamin D deficiency, that clock actually slowed down. So the people's biological age calculation based on all this DNA methylation slowed down. But when you add those vitamins to people who already have normal levels, do not.
Yeah.
So seems like what I understand is, because this correlation is very strong, so I can still, with a caution, use the biological age calculation with enriching certain aging markers from blood, from different. So if I enrich from different organs, it's going to be more precise and robust. So it's not just blood, different cell types. Yeah. So I can use it, but I cannot necessarily just go with one blood and just, this is going to be your diet. So this is going to be your epigenetic nutrition. This is going to be your epigenetic exercise. This is your epigenetic breathing and things like that.
I think it's definitely one of the measurements. It's a very strong correlation. And as a clinician, as a scientist, we tend to measure things, and that's how we want to associate. We want to measure a phenotype, and we want to see that we are improving that phenotype over time. For diabetes world, maybe it's the weight circumference. It's one of those measurements, I think. And we measure multiple things. I think this can be one of those measurements. And once we understood the weaknesses of these things, and then we can definitely use it. It's as a strong scientific basis, at least.
Great. And before finish, I'm very curious. So you're leading the National Institute of Health Morpho Consortium. So, and to me, it sounds like a crazy ideas, but we like crazy ideas in science. So what are you guys doing there? So what you're achieving what you're hoping to achieve at the end? So this is like a consortium. I know there are many other groups involved in epigenetics. So what is this work is about? So
I want to take a step back. So this consortium project. So we know that Human Genome Project, which lasted for 15 years, and they spent about $3 billion. They wanted to reveal the map of human genetic information. That took a very long time. Of course, during this time, the technologies have evolved. And that information took more than 15 years, $3 billion. We can literally now reveal that information in 24 hours with almost $100. So,
wow.
And the reason for that advancement was because of these consortiums that also leads to the new technology development. We were hoping that once we reveal human genome information, we will be able to understand the causes of many diseases. But one thing immediately that was revealed was that it is much more complicated than that, because we understood that the epigenetic information is very important. So then they said, we have to have a new consortium. We have to map all the epigenetic information. That's where I was starting my postdoctoral work at Harvard. That's where the group that I joined was one of the centers. We were mapping human epigenetic information. We were hoping that this will be the new frontier, and it did become a new frontier. We mapped all this information. It revealed new technologies, and I contributed to some of these technology developments. But we also understood that now we know all this information, how physically human genome is organized in every cell. The new thing is that we still don't understand the function of all these human genes. So in our human genome, we have 20,000 genes. Believe it or not, if you look at the PubMed where all this scientific information is being published, only 5% of the genes have been studied. So all this fuss, all this scientific knowledge is about 5% of the genes. Of course, we scientists, we were raising this concern that we need new ways to understand this. So NIH came up with a new big consortium idea. They said that we should develop technologies. We should understand the function of every human gene, not just 5%. So my proposal was one of those proposals. So we are now part of this consortium called Morphic Consortium. We are using technologies to rapidly remove genes and study what they do. And we are doing in stem cells, which means that we can also differentiate these stem cells into different kinds of cells. So the goal is to understand the function of every 20,000 human genes. I think that information is going to be massive. Then I think we will understand the causes of all these diseases in a much clearer way, in a higher resolution fashion. The phase 1 of the consortium aimed 1,000 genes. So we are now in the 3rd year of this. But the second phase of consortium, where we are going to target entire human genes for all 20,000 genes. So I think it's very exciting times. That will also feed into all the epigenetic maps and genetic maps as well. And hopefully, we will be in a much better situation, a few years from now. And when we have this kind of conversation, we'll have a lot more information and we'll speak with more confidence.
Awesome. That's great. I mean, this is very exciting. So basically, 95% missing that we're going to understand at the end of this project. Yeah. And I know you're part of the Human Genome Project with the Eric Lender and the Broad Institute as part of it. And even that's very exciting. So now how we advance from 15 years, $3 billion to just 24 hours and $100. So, and, we know that this regulated metabolic health takes 10 years of your life. So in here also we try to do this comprehensive, but just scientifically based therapies, treatments, diets, exercise, and all kind of counseling and all the interventions that we try to first give that 10 years back. So that's the real longevity. But the actual, even more exciting things will come from those things that we will understand, and maybe from all the screening tools and libraries that maybe we will understand maybe certain compounds that we will have those kind of recipes one day that we can say, if you have this information, use this pill or take that or live in this way, or you will have this kind of risks that you need to be aware of. Yeah. We're not there yet, but we are very excited that you guys have started that. So good luck.
It's exciting times. Thank you.
Thank you so much for being with us today in Istanbul.
It's my pleasure. Yes, absolutely.
It's really an honor to have you.
And congratulations for this center. I hope to hear good news from this.
Thank you so much. So thank you so much for listening to us, and we will continue our exciting scientific talks. Again, we are at the Florence Nightingale Hospital in Istanbul, so we're very thankful to Demiroğlu Bilim University for hosting this seminar series, Flowell Scientific Wellness Talk Series. So we're going to continue every month. And thank you so much for everyone listening to us and our guest again, Professor Adli. See you next month.
It's been a pleasure.
Again in Istanbul.